Hello, today we're going to be talking about cell transport. Cell transport is the movement of materials into and out of the cell via the cell membrane. The main topics we'll be going over in today's video are active versus passive transport, types of active transport, and types of passive transport. So cell transport can be divided into two main categories: active transport and passive transport. Active transport requires energy to power the movement of molecules across the membrane, while passive transport does not require energy for the movement of molecules across the cell membrane. Now, why do certain types of transport require energy while others don't? This question can be answered by looking at something called a concentration gradient. In a concentration gradient, there is a region with a high concentration of particles or solute and an area with a significantly lower amount of solute. These two regions may be separated by a layer, like the cell membrane, which in this case is permeable to the solute. Now here you'll see I've added these square-like brackets next to low solute and high solute. These simply notate concentration, so they read as low solute concentration and high solute concentration. In the gradient, the solute in higher concentration will move to the region of lower concentration in order to reach equilibrium. In passive transport, this movement of molecules does not require any energy expenditure, because molecules are moving from the higher to lower concentration, which is the natural behavior. For active transport, however, energy is required as solute is moving against the concentration, powered by ATP, as shown in this example over here, since the solute is moving from a low concentration to a high concentration, increasing the concentration in the higher concentration region. Hence, this energy is required because the solute is moving against its natural course. Active transport may also occur when a bulk transport is occurring. And now I'm going to be talking about active transport. So there are multiple types of active transport, but the main two categories that we're going to be going over in this video are endocytosis and exocytosis. In endocytosis, large amounts of molecules, or large molecules, are brought into a cell through a vesicle, and in exocytosis, large amounts of molecules, or large molecules, are released out of a cell through a vesicle. And these vesicles are formed by pinching off part of the cell membrane, and that image will become more clear in just a couple seconds when I show you the process through some drawings and walk you through endocytosis and exocytosis. But also, just on a side note, an easy way to remember the difference between the two words is if you look at the first two letters of each word. In endocytosis, the EN, and for exocytosis, the EX. They kind of help you remember the EN for enter and EX for exit, so you remember which is which. So here you can see that I have set the dark teal color to represent the cell membrane, the light blue shaded region to represent the inside of the cell, and the pink dots to represent large molecules. And so what you can see happening here right now in endocytosis is that the membrane kind of curves in and forms a little dip, and the large molecules come inside the dip, and then that dip sort of cleaves off from the cell membrane, and so the membrane of that vesicle is actually taken from the cell's membrane itself. And then you kind of see the exact opposite thing actually happening for exocytosis, in which the vesicle kind of edges its way up to the cell membrane, attaches there, and then it opens up, and it has, at that point, the membrane of the vesicle is attached to the cell membrane itself, and so when it opens up, that vesicle membrane has become a part of the cell membrane, and anything that was the large molecules inside that vesicle have just now been released out of the cell. And now we're going to focus in a little bit more on endocytosis, and we're going to be going over in this case just 4 different types of endocytosis, which include phagocytosis, pinocytosis, receptor-mediated endocytosis, and cotransport. So we will start off with talking about phagocytosis. Phagocytosis is defined as engulfing solid particles. It's actually also known as cell eating, because what happens in this process is that the particles, which are represented by these pink blobs, are taken into a vacuole in the cell, and they are basically ingested, and then they are digested or broken down by enzymes inside the cell, and that is phagocytosis. Now moving on, in pinocytosis, the cell engulfs large amounts of liquids such as water into the cell, and it is also known as cell drinking. So here we have the blue shaded region representing the extracellular fluid that the cell is going to engulf, and the pink dots simply representing some sort of particles or solute in this fluid. And here the cell is engulfing the extracellular fluid, and the pink dots are simply molecules that are brought in, as this is a bulk transport. And now on to receptor-mediated endocytosis, in which a molecule, known as a ligand, binds to its corresponding protein receptor on the outside of a cell and triggers the molecule's entrance into the cell. So here we see the cell, and we see this sort of, the pit that forms for endocytosis is actually coated in proteins, and we'll see the significance of that later. And now these orange Y shapes that I'm drawing represent protein receptors, and the green dots I've shown here, as I'm pointing to, are the ones that correspond to that specific protein receptor. And so once the green proteins have binded to these receptors, a coated vesicle will form, as shown here, and these coated proteins aid in shuttling the molecules that have been brought in within the cell. So now let's talk about cotransport. In this process, 2 substances are transported across the membrane and are coupled together. So in this scenario, we're going to be talking about a uniport coupled with a symport, and right now that probably makes no sense, so let's talk about the 3 basic kinds of transport proteins. The first is a uniport, in which simply one molecule goes from either in the cell to out the cell, or out the cell to in the cell. In an antiport, you have 2 different types of proteins going through it, and they're going in opposite directions. And in a symport, you still have 2 molecules, except they're both going to be going in the same direction. So now that we have that cleared, what we're going to be talking about is a uniport coupled with a symport. So in our example over here, we have 2 transport proteins. The one on the left is going to be the uniport, and the one on the right is the symport. So we have already established 2 concentration gradients. So for the green molecules, outside the cell there is a higher concentration, and inside there's a lower concentration. And for the red molecules, it's vice versa. And so as we discussed before about concentration gradients, the molecules will naturally want to go from a higher concentration to a lower concentration to reach equilibrium. But in this example over here, the green molecule is actually going to go through the uniport from a low concentration to a high concentration, and because this is going against the concentration gradient, against the natural course, it's going to require ATP. And what this actually does is it builds up energy. So now what happens, because there's an even more concentrated gradient for the green molecules, the green molecule will now go inside the cell, and it'll go to the lower concentration, and that actually ends up releasing energy. And in the process, it makes it possible for the red molecule to also go in through that symport transport protein, and it powers that red molecule to go against its concentration gradient. And now we're on to our third and final topic, which is passive transport. And just like active transport, there are multiple types of passive transport. The most basic is diffusion. Diffusion is simply the movement of molecules from a high concentration to a low concentration in order to reach equilibrium, as was shown earlier when I was describing the concept of a concentration gradient. So now, as you can see from this illustration here, the solute has reached an equilibrium, or an equal concentration. And here I'm just going to quickly draw in a cell membrane to show the next type of passive transport. So I'm going to be talking about a passive transport called facilitated diffusion. These two blob type things that I'm drawing right now are representing transport proteins, because facilitated diffusion is simply the movement of molecules via a transport protein. And lastly, we have osmosis. So these U-shaped vessels will hold water and solute. The one on the left is what the initial mixture looks like, and the one on the right will be what it looks like over time. This dashed dark blue line I'm drawing represents a partially permeable membrane, which will be permeable only to water molecules and not to the pink solute that I'm drawing. Now, as you can see, the concentration of the pink solute on the left is much higher than the one on the right. And here I'm adding these blue dots to represent water molecules, and what I've done is actually drawn an equal amount on both sides to show that there's an equal amount of water molecules, so the water level is the same. But over time, as we can see here, the water level will increase on the left because the membrane is only permeable to the water molecules, and that's the only thing that can flow through. And this is the process that we call osmosis. Now, on the vessel on the right, I know I've written high concentration and low concentration for the pink solute, but actually what's happened here is that there's that same high amount of pink solute on the left and the same low amount on the right. But what has happened is that there is a higher amount of water molecules on the left that have gone through the membrane, and a lower amount of water molecules than before on the right. Therefore, the concentration on both sides is actually the same, because there's the same ratio of solute to water molecules on both throughout the vessel. And so what we've reached is something called solute equilibrium, and that is the end goal of osmosis. Thank you so much for watching. If you found this helpful in any way, please like this video and subscribe.